my great pleasure to introduce Werner Lanz, who is going to be chairing the session of this first Breakfast and Science seminar for 2023. And he will be introducing our speaker.
It's my pleasure to introduce you to the speaker of today, a dear colleague of mine, Katarzyna Pierzchala. Kasia is the name we know her. It's much easier for us. Yes, so she got her master's degree in Division of Medical Physics at the University of Silesia in Poland. She then came to Switzerland to do her PhD at EPFL in biomedical physics. And actually, it's at the time that we met for the first time, since we were sharing a big office, an open space office, which was called the Jardin Botanique, with very nice plants and flowers. But actually, the nice name was hiding the fact that you were sitting 10 meters away from a nuclear reactor. As you see, we are both still alive. In 2014, she joined as a research scientist the CIBM and LIFMET at EPFL, where she implemented electron paramagnetic resonance spectroscopy. She also developed optical and fluorescence microscopy to study the brain redox and antioxidant states. And she also worked on magnetic resonance spectroscopy of intact living cancerous cells. And her research focuses on understanding the neurodegenerative disease mechanisms using interdisciplinary tools from biophysics, biochemistry, and biology. So that's sometimes the reason people forget that she's a physicist, hardcore physicist. And she's our local expert in biochemistry and especially, in biomedical methods. So it's a very enthusiastic person, as you know, and she's always keen to help colleagues with pragmatic solutions to your problems. And if you don't have a problem, she will find you some ideas. So it's really a great pleasure for me to introduce Kasia for her presentation. So she will present today on systemic and central nervous system oxidative stress interplay with inflammation in a rat model of Type C Hepatic Encephalopathy. Brothers in arms. We will probably know it at the end of the presentation. Yeah, exactly. So thank you very much.
Thank you very much for the kind introduction. Yeah, indeed, we know for a long time. And I'm happy to be here to present to you today my work on the hepatic encephalopathy and the mechanisms which are involved in the disease progression. So let's start. So the small outline, I will talk about the oxidative stress and reactive oxygen species inhaled in the disease, oxidative stress and inflammation in Type C HE, oxidative stress and inflammation detection, so the techniques which have been applied to this study. Then after that, I will talk a little bit about oxidative stress and inflammation and the cellular response at the level of the central nervous system, and I will bring you the conclusion of the whole batch of the study. So let's start with the oxidative stress and inhaled disease. So where the oxidative stress is coming from? Oxidative stress, it can be, let's say, endogenic, which is a common thing which is ongoing in our metabolism, but also exogenic. So it's coming from the environmental stuff. So we can have the UV radiation, we can have ionizing radiation. Ionizing radiation may come from our own medical exams. We have smoking, we have air pollution. So we are exposed from every side to the oxidative stress. So it's something which is with us for every day. So when we have an increased oxidative stress, it will cause DNA RNA damage, protein damage, and lipid peroxidation. So about the oxidative stress, as I said, is one of the components of our everyday life. So why is it a component? ROS, so reactive oxygen species, are small messenger molecules, which are normal components of our signal transduction within the cell, and it's normal during our physiological processes. But the redox homeostasis is maintained by the balance between reactive oxygen species, reactive nitrogen species generation, and elimination by the antioxidants. So here I brought like a balance, and on the balance we can see like pro-oxidants. So you have reactive oxygen species and reactive nitrogen species, which play a good role and a bad role, as I said before. But then after that, when we have increase of the pro-oxidants, we have some things which have to work against it. So we have the battle of the antioxidants. We have different types of antioxidants. The antioxidants may be endogenous, produced by our body, but also exogenous, which has to be delivered with food, with the nutrition, etc. So when the level of the pro-oxidants is higher than the antioxidants, or the antioxidants cannot cope the pro-oxidative reactions, what will happen? The oxidative stress will appear. So when the oxidative stress will appear, the cells will get damaged, there will be disability of the energy production. Then after that, all of the lipid proteins and nucleic acids degeneration due to oxidation. And in consequence, we will reach to the apoptosis or necrosis, or depends which system, which mechanic will be launched, and it will cause disease. Whenever the antioxidants can cope and they can overcome the increase of the pro-oxidant system, which is launched, then we will have the neutralization of oxidative stress and then no disease. So how we function normally. Wherever the redox signaling is regulating the very crucial factors of our functioning. So, for example, for the brain, is regulating synaptic plasticity and also immune defense for the whole body and also for the brains. In this case, they are microglia cells which are responsible for the defense of our brain. And if we look, about the regulation of the redox signalings, we can look on this triangle. And in the triangle, we have divided on the three levels. So whenever we have too much of a reactive oxygen species, so the oxidative stress is increased, so it will overshot signaling and then will cause non-specific damage to the cells. The same, more or less the same will happen. The disease will appear when the oxidative stress is too low. So they will shut all of the signaling, a lack of host defense because the immune system will stop to working. And then after that, we will have the pathological compensation. So we have to have some oxidative stress involved in our body, which is necessary for the correct signaling, biosynthesis, and host defense. So if we look on the graphic, so we have to go with the normal distribution and we have to be here. So we have to protect our body to fight the oxidative stress, but also we have to keep the oxidative stress enough strong to function. And so sometimes, if people are taking some supplementation of antioxidants, because they think it is fancy and nice, we have to pay attention what we are taking. Yeah. So, but what is important, brain is one of the most susceptible region of our body to the oxidative stress. So oxidative stress and the health and disease. So redox imbalance and so the oxidative stress is a feature of many diseases, like the cardiovascular diseases, neurodegeneration, and cancer, etc. So, and also is related to the aging. So with the age, increase of the oxidative stress is a common factor and is causing, degradation of our brain. So when we look on, exhausted list of the diseases in which oxidative stress is involved, we have a long list, and this is not exhausted list. So if for our study, we focus on the liver disease and the neurodegenerative outcomes, which are related to the liver disease. So if we talk about oxidative stress and the neurodegenerative outcomes in the hepatic encephalopathy and the other neurodegenerative disease, we have to know that central nervous system cells are really vulnerable to the oxidative stress mediated injury because of their physiological and biochemical properties, high energy demand, and unique activities. So neurons, they generate the highest rate of oxidative stress of reactive oxygen species for their functioning, and they utilize almost 20% of the full consumed oxygen by our body. And majority of this oxygen is necessary to produce the ATP, to maintain the intracellular homeostasis. Then the majority of the neuronal cells are non-replicating cells, although we know that some cells of the hippocampus has a number of the stem cells, but there is always the limit. So therefore, the central nervous system cells are really sensitive to oxidative stress. Neural membranes, they are rich in polyunsaturated fatty acids, which are particularly vulnerable to oxidative stress because they get lipid oxidation, and then after that they get damaged. So modification in the channels of activity, the disturbance of calcium traffic across the neuronal membranes also influence the increase of oxidative stress. So we have one of the main oxidative stress species, ROS, which is a superoxide anion, which is then after that dismutated by a superoxide dismutase to the hydrogen peroxide. When I'm talking about that, I will explain later. When we have the hydrogen peroxide in the presence of the intracellular ion, copper or manganese, so free ions within the cells, and we know that in brain we have quite a lot of the metal ions, it will react with these ions. Whenever it reacts with these ions, it cause so-called Haber-Weiss vicious circle, phantom chemistry reaction, and then it will generate one of the most dangerous and active radical, which is hydroxy radical. And the last but not the least about the central nervous system, the antioxidant defense of the central nervous system is quite low because it possesses quite low level of the antioxidants like ascorbate, glutathione, catalase, or glutathione peroxidase, or vitamin E. So let's go now to the oxidative stress and inflammation in type C HE. So what we know, also what we don't know, and what we want to explain. So we know that hepatic encephalopathy is a severe neuropsychiatric disorder, and it's characterized by the altered mental status, and when it's going to the end of the disease, often by coma. So there are some neurological alteration in HE that are accepted to the result of declining liver function. And in chronic liver disease, it results in hyperammonemia because liver cannot anymore detoxify it. So when we have impaired detoxification of ammonium, what it will cause? It will cause in glutamine accumulation increase in the brain, within the brain. It's a common sign in the hepatic encephalopathy. And then after that, we know that there are some several pathogenic mechanisms which are involved in the hepatic encephalopathy. Here today, I'm focusing on oxidative stress and inflammation. So, but the mechanisms of oxidative stress, how oxidative stress induce inflammation and vice versa, are still unclear in every field in the science. I would say life science. So, but it's really important, and it's related and linked to many chronic inflammatory disease. So if we would talk about the oxidative stress and inflammation, we have to divide two systems. We have the peripheral, so systemic inflammation, oxidative stress, and neuronal. So we start here with systemic. So for the systemic, we have white blood cells, and especially in the neutrophils in our body, which are responsible for the immune processes. So they release a lot of pro-inflammatory cytokines and ROS. So it's called oxidative burst, actually to help us to cope with the invaders like viruses and bacteria and etc. But whenever they are in malfunctioning, they might release it because of the going into vicious circle, and that's why they don't anymore have their function, and they lost the function, and then after that they become pro-inflammatory and pro-oxidative stress. So when they are in excess, they cause endothelial dysfunction and can increase the leakiness of blood-brain barrier. And with this, they can allow even further to crossing of the toxins into the brain. So as I said, we have also like the other part. This is the neuro part. We have the neuro part. In the neuro part, for the inflammation and oxidative stress, we have two major groups of cells which are responsible. These are astrocytes and microglia. When they become activated, there is increased level of the oxidative stress and inflammatory cytokines, so inflammatory factors. So that's why, I would say we designed the study in the way to understand step by step what's going on in this disease at the level of oxidative stress and inflammation. So the study were divided to 5 steps. At week 0, we had the sham rats and bile-aggregated rats, and they undergo all of the clinical biochemistry screening. They had we started with the oxidative stress of the systemic system, so with the blood. At the same time, it was Dunia who measured the magnetic resonance spectroscopy of the brain of the rats. So we had like a starting point, we knew from which we started. And then after every 2 weeks, all of these measures were repeated. But additionally, at week 4 and at week 8, what was additionally done? It was done that we had, the withdrawal of the tissue of the brain to perform histology measures. So to get an idea at the histological level, what's going on at the cellular, with the cellular function. So how we detect the oxidative stress then? So oxidative stress is not easy to detect because I will explain in a moment. So in vitro steady state of the reactive oxygen species range from pico to nanomolar range per cell. So it's not so much. So then lifetime span of the radicals and reactive oxygen species is range from the nanoseconds to seconds. Here I have like a table of the like principal ROS RNS species, and we can see that superoxide anion, the one of interest, because this is one more prevalent in our body. The lifetime is 10 to the power of -6 seconds, and its colic, which is very helpful to our body, as I said before, because of the Fenton reaction, is the lifespan is 10 to -10 seconds. So within this, we are reaching very difficult, I would say, process to detect and to catch. That's why we have to go for like a direct detection of short-lived species with the application of the spin probe or spin trap and electron paramagnetic or electron spin resonance, because these are two names of the same equipment. So how did it start? It was the time where Christina, Dunia, and Veronica, they were doing the measurements of the brain metabolism, and what they detect some abnormal behavior of antioxidants. So they found that the ascorbate is decreasing and significantly, and GSH, at least at the level of hippocampus, was decreasing. Also, but not significantly. So then we say, okay, there was a question: what's going on with the system? Is the brain able to cope with the oxidative stress? Is not able to cope. We have decrease of the antioxidants. It's just, or maybe there is not enough because the antioxidants are fighting with the reactive oxygen species. So we want to address the question of the role of reactive oxygen species in the context of Type C Hepatic Encephalopathy. And then after that, also to validate the antioxidant changes detected by proton MRS and its involvement in the central nervous system oxidative stress in the hepatic encephalopathy of type C. So there I came and I said, okay, Christina, let's do this. So I said, okay, I have an idea. This is also my part of my PhD. I was working on oxidative stress for years. So give me the brains of the rats and I will check it. And I will tell you if I am able to detect something. So there I came with the electron resonance, ex vivo detection of oxidative stress. We use the CMH spin probe, which is permeable to the tissue and cells, and that's how we detect the oxidative stress. So this is just a spectra of the spin probe. And this is a stable radical. So for the longitudinal ex vivo EPR oxidative stress detection, as I showed before, we use the EPR benchtop EPR system. We can use different type of samples. Okay, it's just an example that we can use the tissue, brain muscles, cells line, organoids, any body fluids, so blood, plasma, urine, and any other things. So we have to take it, then after that, we have to mix it with certain type of the spin probe. We have different spin probes which are responsible for detection of different type of radicals. Here is an example for detection of the superoxide anion, because this is the one of more important in cell functioning, as I said before. And then after that, what we are doing, we are collecting the spectra over the time. And then after that, with the accumulation, we are able to, I would say, catch the radicals over time, and from this we can build like kinetic curves, and from the kinetic curves we can recalculate the rate of the production of the radicals. So the rate of the production of the radicals we can see here. So what we found, so what is important, we found already an important difference between brain region. We can see, okay, that the first, I would say, test measurements have been done at week 6. So we have the week 6 of the cerebellum, and we can see here the week 6 of the hippocampus, and we can see immediately that the cerebellum, the basic state, has higher rate of oxidative stress than the hippocampus. The hippocampus is here about 80 at week 6, and here we have about 100. Yeah. So why? The explanation is such that we have to pay attention. Although we use the same amount of the tissue, the same, I would say, more or less, volume, the numbers of the cells are different because, let's say, in the case of a human cerebellum possesses almost 50% of our total number of the neurons and 20% of glia. In case of a rat, we have almost between 70 and 80% of neurons within the cerebellum and about 50% of glia of total brain number. So this is related to this, that we will have higher rate because we have more cells involved in all mechanisms which are ongoing. So this is the first thing, and yeah, it's clear. The other things, if we compare here the brain oxidative stress to the systemic oxidative stress, what is important here, we can see already the change, an important change at the beginning, of the disease development, already at 2 weeks, post-operation. So with this, we can say that the oxidative stress detected in the brain, it's appear much faster than this one, which we can see systematically. So this would appear in the peripheral system. So it's very important information. It means that our brain reacts very fast to this, what disease brings it to it. So also, okay, because I have to announce, say this, that of course, as I said before, we have increase of oxidative stress related to the age, and this also have been detected with the rats. So we can see at some point it's more or less stable, but it's still growing. So with the age, we really face a little bit of increase of oxidative stress. So, okay, as I said before, the oxidative stress, the system oxidative stress precedes the one of the systemic, and is much faster because it's already significant at week 2 post-BDL. What is important, also that we found very good, very strong correlation between blood ammonia level, and then after that hippocampus oxidative stress level, also between the glutamine increase and oxidative stress presence. We observe also very strong negative correlation between decrease of ascorbate and increase of oxidative stress. Similar findings, of course, for the blood. That's when the ammonium increase within the blood, we see an increase of also of oxidative stress within the blood with the peripheral system. So, so here, okay, we detected already the superoxide anion using the EPR technique, but just to be, let's say, reconfirm everything and be sure that what we are observing is real, additional, I would say, histochemical approach was applied. So, okay, here is intraductal tetrazole staining. It's a certain type of the staining which will attach to the superoxide anion. So what we can see here, so we have a hippocampus, which is a very important part of our brain, which is involved in learning, memory, emotion, and social behavior, so all of cognitive function. And we have also cerebellum, which play a vital role in virtually all physical movements, so our coordination, etc. So we can see here, I hope so that you can see here, an increase of oxidative stress, very visible, because you here, let's say for the hippocampus, you have fimbrial hippocampi, the part of the tract part of the limbic system, and we see an increase of coloration, which is a direct sign of an increase of the radical production. The same for the cerebellum. Here we have the cerebellum of normal rat. Okay, we see the staining because, as I said, also in the sham, because this is our function, normal function, the metabolic function and transductor function. We have to have this type of a molecule which is making our brain connective. And then after that, what we see in the cerebellum, we see an increase of the staining in the white matter, granular, and also in the molecular layer. So this reconfirmed the EPR study with the first approach of the detection of the histological detection of superoxide anion. And this, okay, when I've seen already, okay, we have the superoxide anion production, then I say, okay, to get rid of the superoxide anion, we have to dismutate it. And the first line antioxidant, which is dismutating the superoxide anion in our body, is superoxide dismutase. So what was observed is an increase of superoxide dismutase 1 and 2 in BDL rats, yeah? When the disease was progressing, what we could see, very important, for the superoxide dismutase 1, which is normally predominantly cytoplasmic, what we could see is a translocation of the superoxide dismutase into nuclei. Why superoxide dismutase is translocating to nuclei? It's a function, it's a mechanism which allows cell to protect itself against the oxidative stress and oxidation of the DNA to avoid, the modification of the genomic DNA, to still allow the cell to function normally, to synthesize the proteins, to make the cell viable, yeah? So, as I said, we see increase of both of them, SOD1, SOD2. So when we have SOD1 and SOD2, normally it's after the next step when we dismutate the superoxide anion is the production of hydrogen peroxide. So the question was, do we see an increase in the production of the hydrogen peroxide? And then after that, if there is a reaction related to. So yeah, indeed, we can say that we have an increase of the hydrogen peroxide, because what we see an increase of the glutathione peroxidase production. So we can see an increase of glutathione peroxidase production, which has been measured. So I say, okay, now we see glutathione peroxidase production increase, but as I said before, we have the free ion present within the brain. And when we have the free ion, metal ion present within the brain, we can face the phantom reaction in the presence exactly of the free metal ion. So what is happening? Then after that, we have hydrogen peroxide, which reacts with the free ion. Here I have an example of iron, but it can be not only iron, but it can be also like manganese, which is present within the brain, copper, which is present with the brain. And also we know that with some kind of neurological disease, this type of the free ion increase within the brain. So when we have the reaction between hydrogen peroxide and the free metal ion, what is happening? We are producing the very harmful radical, hydroxyl radical. And then after that, what will happen when we have the hydroxyl radical? The first, I would say, nucleobase which will react with the hydroxyl radical is a guaninase. It will become oxidase. And we can see here on the staining, here we have the healthy rat, and here is the sick rat, that there is an increase of a staining, which is related to oxidation of the part of the DNA. So when we have also oxidation of the part of DNA and the oxidation of proteins and lipids, what will form also within the cell is there a lipofunctioning pigment, which is a direct sign of the oxidative stress also. So I would say, like, small conclusion from the slide, that the increase of oxidative stress is not really just due to decline of the antioxidants, I would say, level, but rather response to the oxidative stress increase, and thus the body cannot cope anymore with the number of the species, reactive species, which are produced by the cell and coming from the exogenous system. So okay, this is about the oxidative stress. So we detect oxidative stress, and the other question was, is in meantime also inflammation ongoing? We started from the systemic inflammation, just to confirm that the systemic inflammation plays a role in the disease. So what was done? It was the standard clinically applied Guillemot staining on the first point, just to get an idea if we can see increase in the white blood cells numbers, and we can see over like disease progression there is an increase of the white blood cells number. Just to reconfirm it, to get an idea what the number is, the acridine orange staining was performed, and using nucleocanter, we were able, after the separation of the segmentation of the blood, to get an idea how the numbers of the white blood cells increased, and we can see clearly that the inflammation appear already at week 2 post-op. Of course, here we can see also an increase of the white blood cell count in case of the sham rat, because sham rats also undergo some kind of the surgery. It's not full surgery like for the bile duct ligation, so the inflammation is lower, but nevertheless the rat is open. So we have the normal body reaction to the surgery, which after some time, when the rat heal, is decreasing to the normal value. Also, just to be sure that that the inflammation happens, we can see also a certain type of the reaction of the cells. So the neutrophils, they react in certain way that they will cause a hypersegmentation, as you can see here, and this is like a sign of a chronic inflammation. So going further, okay, we know now that we have the inflammation ongoing within the systemic system, so the peripheral blood system. But there was a question: do we see some things about inflammation within the brain? So the two types of the staining have been performed. This is GFAP for intermediate filaments of astrocytes, and also EB staining. This is the staining which concern the microglia. So what is important? We observe an increase of the numbers of the astrocytes. You can see here increase of the coloration, and we also here at week 4 we can see an increase of microglia at week 4. So everything has been controlled. So we could see an increase in the number of the cells. We can of the astrocytes, also increase of the numbers of the cells of the microglia. And it, just to control that we are not observing like more staining or whatever, the total numbers of the cells in the region of interest was also counted. So once one can see also that we see an increase of the numbers of the cells per brain region, which is related to the inflammatory state. So what is important? Although here we see here an increase of the numbers of the cells. At the end of the disease, we can see there is decrease. Increased numbers of the cells can be related to this, that the brain is trying to fight against the disease progression. And some mechanisms are launched to cure the brain. But nevertheless, at some point when the brain cannot anymore overcome, the cells cannot more overcome the disease, the numbers of the cells decrease because they degenerate, unfortunately. And they cannot anymore play their own function. Additionally to this, just to get an idea, a part of the activation of the cells, we have to know if there is a mechanism launched. So for the inflammation in the brain, let's say, and the function of astrocytes and microglia, it's one of them, I would say, more secreted cytokine is interleukin-6. So what we can see here is an increase of interleukin-6 secretion at every brain region at week 4 with a strong increase. In some cases, exactly when the brain cannot anymore cope and everything, we can see the decrease. But there is an evident difference between let's say sham and the sick animal. So with this, we can see, okay, we see an astrocyte and microglia activation because we observe an increase of cytokines and also an oxidative stress. What is important, we have also ventricle microglia. So they are macrophages, which are like part of our immune system located in the ventricles. And over the disease progression, here I have lateral ventricle, third ventricle, and cerebral aqueduct. So what we can see, if we compare the sham at each case, okay, we have the resident macrophages, but already at week 2 or 4, we can see an increase in the number of the macrophages. So they have been called, to come to the inflammatory place to fight against the disease progression. But of course, when the disease is progressing, it cannot cope anymore with the disease. And at the end of the disease, what we can see is like a clot-like formation of the macrophages. So it means the macrophages, they came together at some point to fight some things, and then after that, unfortunately, they cause a vicious circle. And when they cause the vicious circle, what will happen? The inflammation will increase, and also macrophages as the immune cells at the moment they will produce more oxidative stress because they think that they have to still fight something, and then after that they fight between each other and between our brain. So then after that, as I said, we will also talk about the oxidative stress response at the cellular level. So what we have been observing is a change of the morphology of the cells. Here is the GFA-based staining, so which is it's a staining for astrocytes. What we can see that astrocytes, also on the images you can see here, have a sham. It has many processes and everything. And with the disease progression, we see a decrease of the processes. The length, you can see the length is, they are quite long processes. Within the disease progression, the processes, they are shortening. And also the number, of the intersection between, I would say, with the show analysis, you're doing certain type of rings, and then after that, you're checking how many, small processes ongoing from the principal processes for connection between them and their neurons, and we see decrease of the processes intersection within the time. So whenever we know that's the astrocyte, they enshade all of the neurons around. So whenever the processes, they shorten, the connection between astrocytes and neuron decrease. And also whenever the processes shorten, we know that they will detach from the vessels. And then whenever they detach from the vessels, they not anymore hold the blood-brain barrier, which are supporting the blood-brain barrier. And also they cannot anymore evacuate the toxins because there is no communication. The other one, it's a EBA staining for, as I said before, for the microglia. So whenever microglia are in taking a part of the inflammatory and oxidative stress, so what we can see here, what is happening, the microglia, they change their form. They change their form from non-active at the sham moment to the activated at week 4 and at week 8. So what is happening, the cells, they change their shape, they increase the laminification, and also the perimeter of the cells change. At some moment, microglia are trying to remove all of the debris which are happening during processes of oxidative stress inflammation to clean, but at some moment they become too active, and when with the high activation, they become very deleterious for our brain functioning. So what is very important also in this case, when they become active, at some point they lose all of their ramification, and they become amboid microglia, which are very at the very high inflammatory state, also oxidative state. And we can see with the disease progression that we have more of those activated amyloid microglia. The other response at the brain level is a response of the neurons. Neurons responded quite strongly to the disease progression. So what we could see with the Golgi-Cox staining, we have a significant increase of the soma, so the body of the cells increase at the hippocampus level of pyramidal cells CA1. And we have a decrease of spines, so the connection points between the cells with the disease progression, and a little bit different behavior about the soma appeared at the cerebellum. So this is our brain regional difference. Then the cerebellum purpurial cells, they decreased in the cell soma size. But also, they follow a decrease of the spines. So also they use the connectivity between themselves and between other cells within the cerebellum. So we know that the persistent increase of oxidative stress leads to alteration in dendritic vaporization and spine densities in neurodegeneration. So this confirms neurodegeneration effects within the hepatic encephalopathy. We know that the alteration of dendrites are often exactly associated with the cognitive impairment in mental retardation. Interaction between oxidative stress and immune response have direct impact on the structural synaptic plasticity. So this is very important. And as I said before, the microglia cells are involved in this process. They complement and mediate the synaptic phagocytosis. So, the decrease, and then after that, also the spine sprawling. So let's go to the conclusion. And so in the presented study findings, we can say that pathogenesis of the type C hepatic encephalopathy is at early stage can really already exacerbate of the defense of the brain is compromised, and that's why the disease accelerates with the disease progression. We know now that the oxidative stress within the brain appear before the one which could be detected by in the systemic system, so in the peripheral blood system. We know that the elevated superoxide anion production, and then after that overexpression of SODs in alveolar regions might lead to increase of hydrogen peroxide, and then after that it will have negative impact on long-term potentiation, because long-term potentiation is a connectivity between the neurons. But then after that, what is very important, we need it, and at some point, oxidative stress is necessary to build this connection. And that's why sometimes let's say in Alzheimer, we are also observing in the beginning of disease rewiring. So we have increase of the connectivity, and why, oxidative stress. so then after that, when the oxidative stress is exploding, it's not anymore rewiring, it's dewiring. You cannot anymore make the connectivity, and it's losing the connectivity. Then it's very important, we have very strong and negative correlation between superoxide anion increase and ascorbate decrease. So we know, that suggests that the mechanism might be through adaptive behavior of the cells, and then after that, because of the increased scavenging of the free reactive oxygen species, the increase of levels of hydroxy radical. It's a very important point in many neurodegenerative disease and also in hepatic encephalopathy, because it will cause oxidation of DNA and RNA. So when we have oxidation of DNA, RNA, we cannot synthesize properly the proteins. We cannot synthesize properly, the proteins which are necessary for the cytoskeletal scaffold. And we know that the function follows structure. So this is a very important message. So whenever the structure is compromised, the function of the cell will be compromised, because it won't anymore possess the principal function it has when the cell was born. So I said it right now. So RNA oxidation interferes the translational machinery. So it's necessary to make our body functioning properly. So we should eat healthy and make a sport, have decent life. Although I know scientists are a little bit compromised. So we know that then the systemic and neuroinflammation is associated with the type C hepatic encephalopathy. That SOD it's necessary, it upregulates also as a switch to control microglia activation inactivation. In our case, we see increase of SOD. We see also a significant accumulation of interleukins, so the cytokine within the brain, and it's already visible at 4 weeks post surgery. Okay, we didn't do the histology before, so since we have so strong increase at week 4, so I assume that at week 2 we could already see an increase of the cytokine stored within the brain. So what is also important, that the elevated expression of interleukin-6 has been followed in activated microglia and astrocytes, as I said before. And then after that, it will launch the pro-inflammatory cascade. What is very important about interleukin-6 also, that it might compromise the central nervous system homeostasis and allow for neurotoxic components, because it's alerting at some point the function of blood-brain barrier. So it will increase at some point the permeability, and also from inside of the peripheral system, whenever we have an increase of oxidative stress, also it will cause increase of the permeability of the vessels. Yeah, so we have like two sides problem which might increase the toxin transfer into the brain. Also, we could see the levofloxacin aggregates. So this is a direct sign of oxidative stress, and also levofloxacins they compromise the functioning of the neuronal and cells. So with this, I would like to thank you for the attention. I would like to thank all of the BDL team for all the work they do to explain the disease progression, and I hope so, and mechanisms that we will finally be the first in the world and we'll find the solution. And I thank you all for your attention.